and welcome to the Computomics podcast. Our guest today is a wheat geneticist. His career stops include various at the University of Liverpool. He was the research lead for the Institute of Integrative Biology there, the co-director at the Center of Genomic Research, and the academic lead of the Liverpool Gene Mill. And today he heads the plant genomics at Earlham Institute. Welcome to the Computomics podcast, Anthony Hall.
Hi. So should I start off by telling you a bit about the Earlham Institute?
So yes, please do tell us.
Yeah, so the Earlham Institute it's a BBSRC funded institute. So the BBSRC is one of the UK's big research funders. So, and it's a nice place to work because, if you're working in a university, you've got the pressure of teaching, admin, and research. Whereas at an institute like the Earlham, we just do research. We just focus on research. So it's a nice place to work. And I think, and kind of our area of expertise, or kind of where we fit in, is at that kind of data science and technology development. So really focused around sequencing and interpreting genomic data, but also we're interested in synthetic biology as well, and how we can use hypotheses that we develop from looking at our genomic data to test specific ideas using synthetic biology approaches. But it's really that data science and the technology that generates that data that we're really interested in. And that's kind of our unique point where we fit within the scientific ecosystem. And I think what's really nice about the Earlham is we've got that, we generate the data, we have the sequencing machines that generate the data, and we analyze it. And I think that working together with the technology and the data puts us in quite a unique position.
Can you elaborate a little more on that? How does this closeness help you, or how do you think this impacts your work, having both under one roof? Right.
So sequencing technology is moving at an incredible speed. So, I mean, if we just look a little back in history, so before the start of the 21st century, we had the first eukaryotic genomes and prokaryotic genomes. And then in 2000, we had the human genome. And then there was huge technology leaps in sequencing. So it suddenly became, in around about 2005, high throughput. So before, you used to have a warehouse of machines slowly generating sequence, and it was replaced by just one machine the size of a washing machine. So this kind of step change in high throughput sequencing. And that moved us away from thinking about human genomes or very small microbial genomes to we could start to generate genomes for some of the key model organisms. And then after the model organisms, we started generating agricultural genomes. So pigs, cows, tomatoes, rice. And then as the technology advanced further, we started to go for some of the bigger genomes. So like barley and wheat. So wheat is probably one of the biggest and most challenging genomes that we've played with so far. So compared to a human genome, it's 5 times the size of a human genome. And whereas the genome is the human genome just has 2 genomes, so it's a diploid, so it has 2 genomes in there. Wheat's hexaploid, so it actually has 6 genomes. So though each of those genomes is closely related, so it's a real puzzle to start to pick it about and take the sequence data you generate and assemble it into genomes. And that's really been the drive of my research at the Earlham is sequencing the genomes, but not just sequencing them, using them to understand some of the biology that's going on as well.
I'd like to explore both the data wave that you're writing and how you're doing that exactly, and then also the research results. Maybe since you've just explained how the technology has changed in leaps and bounds to a washing machine-sized sequencing machine, basically, that spits out more data than the warehouse used to spit out. How are you, and I'm quoting you here from one of your presentations, how do you surf the wave of data that's coming along?
Yeah,
how can we envision that?
Yeah, it's kind of getting worse. The wave's getting bigger and bigger and bigger, and I mean, just to go back and talk about the data and the last step of the technology that would take us to where we are now. So I said the washing machine. So there's two things that happened over the last 3 years, and two technologies that have suddenly become they've been developed, but they're now really deployable and starting to have a real impact on our ability to assemble complex genomes. And one of those is the Nanodoll technology. So this generates ultra long reads that we can use to start to stitch together our genomes. And the nanopore, I said about washing machine size. So a nanopore is about slightly as big as a mobile phone. So we're
getting smaller and smaller.
From warehouse to washing machine to mobile phone. And then the other big technology, which is still at the washing machine size, but it generates long reads. So this is the PacBio. And what it does is it generates these hi-fi reads, so really accurate long reads, so 20 kilobases in size, so long reads. And what you're doing is the washing machine size machine previously was generating these really short reads, and now we've got these longer reads. It's like a jigsaw puzzle. So previously, with all of these little short reads, you were trying to make a jigsaw with millions and millions of pieces, but as we're getting longer reads, the jigsaw is getting bigger pieces, and we all know that bigger pieces, it's easier to stitch together a jigsaw. So the problem, the challenge of trying to assemble the genomes is almost a solved problem. Mm-hmm. With it being a solved problem means, and now kind of quite cheap, it means we're getting loads and loads of these genomes. So rather than, I mean, I said earlier that we got to a point where we could now assemble wheat genomes, right? So assemble one wheat genome, but it's not just one. We're now getting multiple wheat genomes, but with this new technology, as higher quality or better than the single wheat genome we originally had. So the problem is you're getting all of these. How can you use them? And I think that the research community and both in the public and private sector, we're at this position where they've just been able to deal with a whole reference genome. It was transforming the way they developed their breeding programs or developed markers or could understand the genes underlying some of their key traits. And then suddenly you give them another 16 genomes, and they've just got to understanding what they did with one, and now they've got 16. And they know they need to be able to use them because they know their competitors are thinking about how they're going to use this data. So I think there's now a real challenge to think about how you can build the tools that allow researchers, so breeders and crop scientists in the public and private sector, so how they can use this data. How can we build the tools for allow them to explore that data and make discoveries? So they need the tools, they need the expertise, so scientists that are able to deal with these data sets, and the capacity, the number of scientists to be able to do it. So I have a real key problem at the moment, which is, so I have postdocs in my group that we spend loads of time learning how to deal with this data, and along comes a agri-biotech company or a small startup or a computer science company, doesn't nick them. They move forward into really good careers within that. So we're generating that capacity that allows us to so we are generating capacity. We need to do it more, because there's a real space here that we need to have the tools, capacity, and expertise to be able to deal with it and make discoveries.
What would maybe, thinking of this problem of grand theft, not grand theft auto, but grand theft postdoc that you're suffering from, do you see a way where maybe AI technology can help you deal with that? Because I mean, obviously I'm asking because it's the Computomics podcast. Computomics does has a lot of products that work with AI technology, but just generally speaking, lots of data, processing lots of data, that's where AI becomes interesting. Is that something you're working with currently, or where do you see possible applications?
I guess there's where we see it. what we're doing at the moment. So I think there's a real interest in how when you have lots of these genomes, how you link these complex data sets with phenotypic variations, so changes in the field, and how you can bring those two together. So you've got the pattern of the genome, and then you've got the structure of the plant or the trait that you're looking at, and how you bring those two together. And I think it's been an area of statistics, but I think there's a real possibility to start to add AI there, and it's a real complex problem. So you've got the genome that's fixed, you've got the phenotype, but you've also got things like the way you manage that crop, and you've got the environment, the soil. So trying to integrate all those data sets sounds like a real AI problem. But I mean, that's not something that my group are really pursuing, but I know that that's a big question being asked in kind of the agricultural AI sector. And I think actually the bits of where we have been working with AI, the first step as kind of as a wheat geneticist or wheat genomics or crop genomics person is to think about the data sets that you generate and making sure that they are computer readable, so a computer can come in and use that to then generate algorithms and use AI to identify the patterns. So I think there's a real power in making sure that the data we generate is what we call AI ready. So it feed in. And then there's also to think about when you are generating data sets, could you generate the perfect data set that any machine learning AI expert will go, yes, that's really exciting. And they'll come and they'll start analyzing your data and developing algorithms on your data set. So actually they're making and developing the algorithms that then you can use to make the biological discovery you want. So I think that's quite a challenge. And that's something that we're thinking about in some of the new proposals that we're writing is to build data sets that we can engage with the AI community to start to use to address specific questions that we're interested in. So that's one way of AI. But we are as a group, we are using AI, but in something quite different. So we're really interested. So having a genome is interesting, but there's a whole other levels of complexity. So a genome just provides the code. What you want to add on is models of where the genes are. So they're the bit that kind of drives the biology, but also you want to understand how those genes are regulated. So in a genome, you've got something called the promoter or the regulatory region, and understanding how that regulatory region drives expression of that gene. I think it's a real challenge, and that's something we are using AI for, to try and see if we can identify patterns within those regulatory regions that can then be used to predict how a gene is expressed. And that allows us to start to kind of decode this part of the genome that is currently not accessible. And it's really, really important to start to understand that region, that coding region, that non-coding region, because if you go and look at data, for example, in maize, around 40% of the inheritable genetics within the driving traits is in this non-coding region. So actually, if you can understand the impact of a mutation in the non-coding region, then you can work out whether that's the cause, that's the reason why we're seeing that phenotype. At the moment, it's more straightforward if it's a coding region, because that mutation causes an amino acid to change, and that's not an AI problem, that's a simple biology problem. But understanding that regulatory region, there's a real need to develop tools around that.
I mean, with 40%, that's a sizable part of the area that is what you term non-coding. So I can totally see that where AI might help.
Yeah.
You said you don't work with AI that much, but then you proceeded to name quite a few key areas where you work with AI. Can you share a couple of the research results that you have gotten in that research? Not necessarily just with AI, but overall.
Yeah. So I'll talk about the AI stuff we've been doing. So we had a paper in PNAS last year, and that was in collaboration with IBM. So, and actually it was in collaboration with Laura Gardner. So she was the lead author on the paper, but it was a collaboration between IBM and my group. And so Laura was previously a PhD student, then a postdoc in my group, and then moved to IBM. So I would say they nicked it. They didn't nick it. She's on a similar salary to me now. So it was a good move for her, a really good career move for her. So in that paper, what we did is we developed methods for using data to identify biomarkers that allowed us to. So one of the data sets you generate is gene expression data. And what we wanted to do was use this gene expression data to identify whether we could use gene expression data to identify a complex trait. So we're one of the things that we're interested in. So I'm interested in wheat genomics. We're also interested in biology, a biological process, and that's the circadian clock. So how plants tell the time. So this is an important agricultural trait, but it's really difficult to measure in the field, because basically to measure it, you have to be able to assay the plants every 2 hours for 48 hours under constant light. You don't get those conditions in the field. So what we did is we developed a machine learning algorithm that allowed you to predict the internal time from just a single transcriptomic time point. So basically, rather than having to sample over 48 hours, you just take a single transcriptomic time point, and it'll tell you exactly what the internal time of the clock was doing. So in that way, you can see whether the internal clock, whether it's in sync with the external light dark cycle, or whether there's a desynchrony. So it's a way of, rather than generating these huge data sets, taking just one time point and measuring this complex trait. And that's incredibly valuable, because now you can go back to all of the data that already exists. So loads of transcriptomic data from lots of different conditions, take our set of biomarkers and go and ask, what time of day did it think that plant was growing at? So that becomes incredibly useful. And what we did, well, there's one kind of cool experiment we did was, so we thought, this is brilliant. There's an Arabidopsis project called the 1001 Genome Project, where they sequenced 1001 Arabidopsis genomes. They also generated transcriptomic data for 600 of those plants. So we can go in, use our biomarkers, and find out what time of day their internal clock thought it was. Mm-hmm. And we thought, this is brilliant, because we can do that. We've got the genetic data, we can measure this complex trait, and then we can do a genome-wide association study and identify marker trait associations with our new biomarker set. But it turns out that the group weren't that careful with how they designed their experiments. And when we analysed the data, we worked out that the technician came in at 9 o'clock every morning and left at 4 o'clock. So we didn't get, we could work out what time of day the experiments were taken, but we couldn't use it to see whether there's a desynchrony between the internal and external clock, because the external time wasn't recorded. And I guess kind of the take home from this is, make sure your data is you record everything with your data. If they would have said exactly what time of day they were harvested, we could have used that data. Mm-hmm. So luckily, there's another group led by a guy called Magnus Norberg, and he was working on a similar collection, a sub-collection of the 1001 Genome Project, and he actually did record exactly what time he measured. He took his samples, so a specific time in the morning. So we've gone on and we're doing that analysis now and using a genome-wide association to identify the genes driving the variation in this trait. So that's an example of how we've used machine learning to look at a complex trait. and that was very focused on Arabidopsis. But we can now start to move that into crops now. So use the same process to develop markers in wheat, and then we can then take that into the field and see whether different field conditions or environments change the time of the clock just using single transcriptomic measurements. And the same process, you can start to think, well, actually, there's the clock. We can then go and start to look at other complex traits beyond just the circadian clock. So you develop a set of biomarkers. And this isn't really that clever. It's what they're doing in medicine already. So quite a lot of startup companies are developing biomarkers for ability to recover from cancer and things like that, or whether you've got cancer or not, or whether you've got this disease or that disease. So by taking single transcriptomic data sets, they can identify biomarkers within that and using machine learning. And then using that, they can kind of identify phenotypes or traits or diseases within humans. So it's the same idea, but then translated into the agri-tech sector.
That's amazing. I think in what you've just explained, how you are able to use machine learning to develop methods to decrease the data you have to deal with, and then find complex markers or complex traits and identify them. And even I'm also interested in this meta level of going back to this idea of creating data sets that can actually be used for that are AI ready, that you can use for further analysis. And if you fail to record the time that you're actually doing the work, then that might actually be almost like a dead end street as far as further analysis is concerned. So there's almost like two results from that experiment you just described.
Yeah, and it really shows you the power of really being careful about how you design your experiments and explain your experimental data. Because if you're careful about doing that, it means that it becomes reusable. And, you've spent a lot of money, a lot of time, a lot of effort doing that, generating that data set in the first place. So why not make it as reusable as possible? Because ultimately, as scientists, we want our work to have impact. And if our data sets can have a life beyond the experiments that we designed for, that's absolutely fantastic. I guess that's almost my lab motto, is we kind of, we like to use other people's data sets to ask questions that they didn't design their experiments to ask. that's kind of where we go.
Mm-hmm. But you would say you are asking people, please don't just capture the data, capture the metadata. Would that be a fair way to summarize that?
Yeah, and also, and there's that value of thinking how your data might be used in the future. So going and talking with, having a chat with people that are doing machine learning and thinking about how they might like you to design your experiment so it will have maximum impact. I think it is incredibly valuable. I think so many times, we see data that we think is perfect, and then that they're just missing some key component that means it's not useful. Mm-hmm. So maybe I could talk a bit about, I guess the other thing that when we've talked a bit about AI and a bit about how we're using that to look at genomes. I guess the other thing is that some of the stuff that having multiple genomes is starting to reveal that we have no idea about. So I could maybe talk a bit about that. So
please.
Yeah. So one of the things, I'm a wheat geneticist and a wheat genomics researcher. So one of the things that we kind of thought was, wheat is a complex genome, but there's arguments about when it became a hexaploid. So I think there's some papers say 1.5 million. There's some say 10,000 years. But it went through a genetic bottleneck, because it became a hexaploid that only happened maybe once. So the fact that it came through a genetic bottleneck means there's not, that you would expect there not to be that much variation, but yet there's a lot of phenotypic variation in wheat. So that's kind of been one of the things, why does it have so much genetic variation? And I think that's one of the things that having full high quality genomes, not just from a single reference, but from multiple references, is revealing. So what's happened, what we think that has been happening is, when you go and have a look at a wheat field now, you'll see it, and it's all perfect and everything looks the same. But that's a modern wheat field, not an ancient wheat field. So 150 years ago, the way wheat would have been grown would have been as what we call kind of a hybrid swarm. So not just bread wheat, but a whole series of closely related grasses. And then you'd run your scythe across it, harvest your wheat, and then take it to the mill and grind it. And then, you would keep some of that grain and plant it the next year, and that's the way that it kind of works. So you'd always have this a hybrid swarm of a mixture of different grasses. So what's been happening since the hybridization event, maybe tens to hundreds of thousands of years ago, is that you've been getting hybridization between these wild relatives. So actually, the wheat genome is a mosaic. And that's what we've kind of found by generating these genome assemblies, is that, there's the bread wheat genome, but actually there's lots of introgressions of these wild relative bits of DNA all over the genome. And that introgression is giving you the variation, or at least some of the variation we see within wheat. And it is controlling some really important traits. So we have a paper that's currently on bioRxiv that's under review, and it's a collaboration between ourselves and CIMMYT. So CIMMYT's the International Wheat and Maize Research Center in Mexico. And we did a genome-wide association study looking at heat tolerance. So we basically grew our plants we grew a panel under optimal conditions, and then grew a panel that we planted late, so it became heat stressed. And we identified a number of marker traits that actually increased yield by 56% under heat stress. So a huge increase.
Wow.
Right.
56%.
56% compared to with or without the alleles. So it's a really big impact. So we got it down to a loci on one of the loci on 6D, so one of the chromosomes of wheat. And then because now we've got the wheat genome and we've got genomes from close wheat relatives, we're able to identify that actually where the marker lay was in where there'd been an introgression with a wild genome called Taoxai. So we could see that actually the thing that was giving the heat tolerance wasn't the bread wheat, but it was this introgression of this Taoxai region. And we could look through all of the lines, and all of the lines that had this tolerance had this Taoxai bit. And you could then also look at those lines and see where the breakpoint between wheat and Taoxai was in all of those lines, and slowly decrease the region where we thought the gene was. And we're now down to 1.6 megabases, right? So there's still a lot, 1.6 million base pairs. But if you think that the wheat genome is 16 billion base pairs in size, then we're pretty close to identifying the gene. And what we can do is we can go and reconstruct exactly what that interval looks like, because we've got the Taoxai sequence now, and we can see that there's some novel recombinations between genes going. There's some genes that have point mutations that are likely to affect the function of some of those proteins. So we've got a whole set of candidate genes we can now go and look at. And it's a really important trait. And where we're going with that now is we're looking to see whether, okay, so it has this effect in Mexico. So what happens when you run climate scenarios based on global warming? So could this marker trait be important for not just Mexican wheat varieties, but European wheat varieties? And will it help to mitigate the effects of climate change? So that's what we're doing at the moment, trying to introduce that into European lines and see whether it will be useful for breeding programs. Mm-hmm. But it's all been driven by by some elegant physiology and field phenotyping, but also the genomics has allowed us to really nail down the reason that it's actually a wild relative introgression that's the responsible for it. And I think as we start to unpick the complexities of the wheat genome that you getting from these data sets, you'll see a lot more. Well, we are seeing a lot more of these introgression events all around the genome. And these are likely to be linked with a lot of interesting traits to do with biotic, so resistance to various pathogens, and abiotic stress, so things like heat and drought. So there's a lot of useful variation that's going to be incredibly powerful going forward. And we've now got the tools to start to uncover it. So it's all about, right, we've now got multiple genomes. We need the tools for the breeders to be able to use this data to do the kind of experiments we've just done with the CIMMYT material. So we're at that point where, we need to put in hands the expertise and the tools for them to be able to analyze it. And that's data science and genome analysis rather than AI. But as we start to, we can do this by hand, not by hand, but by computers. But as the data sets become bigger and bigger and bigger and bigger, there's no way we can do that. We're going to have to think about applying AI approaches to start to unpick some of these complexities.
I mean, that's amazing. Those kinds of results, but also the outlook you just sketched of being able to identify these key markers, and especially with looking at climate change, it's like that we're going to need it sooner rather than later. So is that the what you're working on right now, to model these climate scenarios with European wheat lines?
So it's a grant proposal that we submitted 2 weeks ago.
Very fresh off the press.
But yeah, I mean, we've been talking about the idea, and it's a collaboration with the University of Nottingham and with CIMMYT and with the University of Bristol. So the University in Bristol have got a lot of expertise in modelling kind of future climate scenarios. So basically, we're providing the genetics and the genomics component of it. Nottingham are the physiologists and run these carefully climate controlled cabinets. So you can run a growth cabinet to mimic exactly what the climate's going to be in 2050 or 2080. And then we can then put in our plant material and see whether these markers are able to cope with these increase in temperature or so increase in temperature, but you're also going to have these what we call increase in these severe weather events as well. So we can program those within the cabinet as well. So you suddenly get a real high heat wave for a week or so. And would the markers help us? Or the traits we've identified help us with those kind of scenarios as well?
That's very exciting. If someone would like to follow up on everything they heard from what you're working on, where can they find your work, or specifically what your group does?
Yeah, so if they want to find out, I mean, the easiest way is to just Google Earlham Institute. Anthony Hall Earlham Institute, and they will find me. That's the easiest way. And on that site, there's email addresses and things as well. And we're always looking for talent. So
that's given the grant theft. I'm just going to keep calling grant theft postdoc. I've kind of fallen in love with that term now. So you're looking for new talent. Can you maybe, if someone's listening right now that is interested in that field, what would you like to tell them?
Yeah, they should get in touch. We can discuss stuff. And what we're kind of keen to do is we're really happy to take people that have some computational biology skills, but we're happy to help them develop those. So they too can get a job at IBM and twice the amount that I do. But yeah, I do think that there's some really interesting science in this area going on at the moment. But also, I think if I was 25 now, I think it's an area where you could come develop some skills and then your career would be sorted. I think that there's a real demand for talent in this kind of computational area. So,
well, that, and I think specifically dealing with with those talents in that field of, it's basically about trying to eliminate world hunger, even with the climate change that's upon us already, and that will probably just keep getting worse. So it's something where your work will be quite impactful, regardless of whether they keep working with you or decide to move their talents to the industry maybe. But it's definitely something where any research results or any advancements that would be produced as far as breeding plant biology is concerned would really impact all of us every day almost.
Yeah.
It's an exciting outlook. Well, Anthony, thank you so much for taking time out of your day to tell us about the research you've been doing. I thought it was really exciting to learn, especially with the example you gave with this experiment of the heat exposure, the heat stress, 56% increase, and how being able to identify that Tau shy, I think you said.
Tau shy, yeah.
Tau shy.
Eight drops Tau shy.
How basically that wild piece of wild relative DNA within the genome really was responsible for that, and how the different data sets helped you identify that. I think that was a really great example to illustrate what you explained before, and how the data sets kind of work together and what we need, but also how complex it is to now develop tools and have enough people and the expertise to really move that basically to implementation on the breeder side. So that's definitely something we'd like to keep an eye on. And for our listeners out there, Anthony said you can just Google it, but also we will be putting up a link in the notes on our site, computomics.com, so you can explore that a little more. Once again, Anthony, thank you for your time, and to everyone else, hope to have you back next time for the computomics.com podcast.